Hey everybody, I studied the transcriptional regulation of the plant immune receptor NLR genes in immune responses. The clicker isn't quite working, so I'll just do it manually. Okay, so NLR proteins recognize pathogen effectors and lead to cellular response. Sorry, this isn't working. Can I have some help with this? Just the clicker.
No, that's the point.
Yeah, it also lets you click though.
I guess maybe just use the computer for now.
Okay, do you know if the pointer works for it?
I'll see.
Okay. Okay, thank you.
Work now.
Okay, so I'll just restart again. So hi, I'm Evan, and I'm studying the transcriptional regulation of plant immune receptor NLR genes in immune responses. So, slide. So intracellular immune receptor NLR, or nucleotide binding leucine rich repeat proteins, have evolutionarily conserved domains in the central and C-terminal domains. However, the N-terminal domains allow for some variety in these proteins. So continuing onwards, the function of NLR proteins is to recognize pathogen effectors and lead to cellular responses. As you can see up here, effectors enter here and they are recognized in the cytoplasm by the NLRs, and this leads to NLR activation and ETI, or effector triggered immunity. So our study is about how the expression of NLR genes must be controlled. So SNCC1, which you can see right here, is an example of an NLR gene that leads to increased immune response, and it is activated in response to pathogen infection. BON1 is a protein that serves to repress SNCC1 expression. And when BON1 expresses or represses SNCC1 expression, you end up with a Columbia or wild type phenotype, which is shown on the right. However, if you have a mutation in the BON1 protein, and this leads to SNCC1 being able to have increased expression, what happens is you have a stronger immune response. And this can lead to the BON1 autoimmune phenotype, which is shown on the left. The reason that this happens is because energy can be taken from other life processes, such as growth and reproduction, and be moved to support the very, very strong immune response. So as you can see, the BON1 plant is much smaller than the Columbia plant. And in the context of crops, having an autoimmune phenotype would mean that you would get much less yield for your crops and you would get much less food consequently. However, on the other end of the spectrum, if you have too little of an immune response, your plant would become more susceptible to things such as pathogens, and these plants could die. Consequently, it is important to balance the amount of immune response in plants when trying to maximize the yields. And by studying NLR genes and their regulation, it is possible to tailor plants towards their specific microenvironments so that you can increase crop yield. And that is the end goal of this project and many other projects that are associated with this project. Okay, so while BON1 is a regulator of the SNCC1 gene, there are many others. And we, as the title of this project suggests, we hope to study the transcriptional regulation of NLR proteins, such as SNCC1. So what we have up here in this blue box is a locus for the SNCC1 gene. And down here, as shown by the titles WRKY 15, WRKY 18, WRKY 22, WRKY 28, WRKY 40, and WRKY 75 are blown up regions within the SNCC1 locus that may reveal locations where there's increased binding between transcription factors and DNA. These areas of increased binding appear at these peaks, for example, and we can use these to determine possible locations of transcription factors that may or may not elucidate the location of WRKY transcription factors. However, to be fair, the DAP-seq analysis is performed in vitro, which means that it's done in a test tube or not in a real life situation. So we don't know for sure whether or not these are transcription factors. So in order to test out whether or not these regions have effects in regulating SNCC1 or NLR protein or gene expression, what we are going to do is we're going to take tDNA inserts, knock out the region, and see what effects occur. So before we see the effects of tDNA, mutants like tDNA knockouts to see the gene function, we first have to have a way to see the possible effects of these. And what we do is we generate BON1 worky mutants in order to help this visualization occur. So we previously discussed that BON1 suppresses SNCC1 function. And this leads to increased R protein expression, which is similar to what occurs in pathogen expression. So when we have the formation of BON1 worky mutants, we are more able to see the effects of the transcription factors. And the whole point of this is using the BON1 mutant background allows us to correlate, or it theoretically allows us to correlate growth phenotype with SNCC1 expression. And the more SNCC1 expression would make us theorize that there would be a smaller plant. So moving onwards, we derive the BON1 worky double mutants and even triple mutants by performing crosses between lower order mutants, such as a single BON1 homozygous mutant and a worky mutant. So what you do is you cross the two plants to get an F1 generation, and you allow these plants to self-cross to get an F2 generation, which should consist of 1/16 BON1 worky homozygous double mutants. Based on this, we next have to identify the double mutants so that we can locate them and study them more. So this is how the right side of the slide is how we do this. So if you look up here, you will see there's regions called the LP region and the RP region on a regular strand of DNA without the tDNA insert. So what you can do is you can attach primers to the LP and RP regions and duplicate the region in between using PCR, and then you can measure the locations, on a gel. And based on doing that, you can see whether or not the locus is visible. However, if there is an tDNA insert in there, what would happen is the length between the two would be increased greatly, and you would not be able to see DNA replication or a band on the gel. This is why if you were using the LP and RP primers, you only really see a band if it's wild type or if it's heterozygous, because one of the strands does not have the tDNA insert. However, in order for the LB band and the RP band to result in DNA duplication and to see the bands for these, you must include the tDNA insert. So that is why there's only bands if it's a heterozygote or a homozygous mutant. So just to show you, this is a little bit of a hard concept to grasp. So we just want to walk you through a real life example of this. So this is a picture of one of my gels right here. And on the right, there's worky 18 genes, and I use the left primer and the right primer, LP and RP. And if you look at lane 1, for example, there is a band right there. There's a pretty strong band. And there's not a band right here, which would indicate that for the worky 18 gene, it is ++ or a homozygous wild type plant. However, if you look at another lane, such as lane 5, where there is no band, and on the LP and RP, and with the LB and RP, there is a band, which would indicate that it's a homozygous mutant. And when we're trying to generate mutants, that is what we look for. So now that I've walked you through how you screen for and generate these plants, we're going to see the results of what worky 15 and worky 22 do. And what they likely do is they likely repress SNCC1 expression. So as you can see on the chart right here, we are comparing BON1 with several of the BON1 worky double mutants. And for the two samples here with the stars right here and here, there is a significant difference as calculated using a t-test. And we see that these plants are actually smaller than the BON1 plant, which means that the BON1 phenotype is amplified. Because of this, we know that it is likely that more SNCC1 expression occurred, which means more of the autoimmune phenotype was observed. And these plants are smaller, indicating that worky 15 and worky 22 likely repress SNCC1 expression. And just so you can visualize it, this is worky 15 and this is the worky 22 double mutants, and you can see that they are by far the smallest on the slide. We also performed this analysis using worky 18 and worky 40, which in prior literature were hypothesized to have partially overlapping functions, which would mean that you would have to knock out both genes in order to determine the functions. So if we go to the previous slide on how we generated the double mutants, you would just do the same process again, except start with a BON1 worky double mutant, and you would just perform the same process to get the triple mutant. So here we see that the bond 1 work E18 and the bond 1 work E40 plants are similar in size, while the bond 1 work E18 work E40 plant is much smaller. And we see that these two plants are similar in size to the bond 1 plant, while this is much, much smaller. So when we ran an area-based quantification, we found that the bond 1 work E18 work E40 plants were much smaller, indicating that this promoted the bond 1 phenotype, both of the transcription factors, and that they had overlapping function as expected. So here are my conclusions. I just stated them. And our further research is to perform pathogen growth assays on the worky mutants, because we previously used a worky background to try and see if there is more SNCC1 expression or if there is a change in the growth phenotype. But this is just a substitute that's used to make the process quicker and screen more plants. We need to do a pathogen growth assay for increased accuracy and to see what actually occurs during pathogen infection. And we also want to check SNCC1 gene expression to make sure there are not other extraneous causes that are responsible for the changes. Okay, so that's the end of my first project, but here we go, move on to my second project. And in this project, we know that not all NLR genes are like SNCC1. Not all of them are upregulated in response to pathogen infection and cause increased immune response. So this is a project that my wonderful mentor, Leun Yang, helped me with. So originally she started and she created a chart that analyzed all 207 NLR genes in the Columbia accession. So what we find up here is that the vast majority of these plants are upregulated in response to pathogen infection. However, a few are actually downregulated in pathogen infection, and they are shown right here. So my goal was to take a few of the samples that Leun did not get the best results for when she was trying to figure out the pathogen infections for, and to try and find out more, like what are the possible functions. So I did some deeper analysis of this by running some pathogen growth assays. So on this slide, you can see the result of some of the pathogen growth assays I ran. A pathogen growth assay is basically where you take the plants, you grow them, and some of the plants, such as 1, 2, and 3, have tDNA knockouts, that were on the previous slide, and these are the genes that are knocked out. And you basically analyze how do the plants respond to infection with pathogens. You calculate a value that represents how many bacteria are there per unit area or per unit mass in the plant, and that tells you how strong the immune response is within the plant. So I performed a few pathogen growth assays, and if you look at the areas in yellow, those are my significant results. And my significant results are shown right here with the stars, and they illustrate that the areas where there's a significant change in the number of bacteria observed as compared to the wild type were all decreases in the number of bacteria observed. This means that there's actually a strengthening of the immune system, which indicates that some NLR genes actually repress the immune response. And this would make sense because you would want to turn off genes that repress the immune response. So it makes sense why these genes are turned off. So moving on to my conclusions, I previously stated that some NLR genes are downregulated in response to pathogen infection. And because of this, we noted this trend only for 3 plants. We want to investigate more plants, and we want to perform more repetitions to see whether or not this trend holds. Thank you very much. And here are some acknowledgements. I'm especially grateful to Leun and Professor Hua and the other members of Hua Lab for putting up with me and all the mistakes I make. And I really love their support, their enthusiasm for their work, and just learning about what they did. And it inspired me to go and join the lab for next semester. So thank you very much.